Hello, everyone. I am Asif Parker. I work at SwanBio. SwanBio is a small biotech company based in Philadelphia, where we are developing AAV gene therapy for AMN. This has been an informative event for me, and I want to thank Sarah, Karen, and their team for organization. And thank you for inviting me. It's a true pleasure to speak with you. Today, first, I want to give you a short introduction to gene therapy and how gene therapy approach works for AMN and how it is different than the inflammatory cerebral phenotype, then I will share the findings of animal studies depicting the efficacy of AAV approach. If we go to the next slide. Genes are made up of DNA, which are blueprint to build proteins that make our body work. For example, ABCD1 protein is built by the gene affected in ALD, and this protein helps breakdown of very long chain fatty acids. Gene therapy introduces a working version of the faulty ABCD1 gene into the cells, the gene that is in charge of creating ABCD1 protein. This is done by using a vector, which is often derived from a virus, but the viral genes have been removed. Only therapeutic genes are delivered into the cells. The modified cells then produce the protein that was missing or defective prior to the treatment. The vector can either be delivered outside of the body, that's ex vivo approach, that's depicted on the right side of the slide, or the vectors can be injected into the body, directly into the body. In vivo treatment, that's on the left side, sorry, that's on the right side of the slide. The ex vivo approach is used to treat the cerebral form, the Bluebird Bio's gene therapy. Blood cells are removed via blood draw, and you can see it here on the left side of slide. Blood cells are removed. Then the stem cells are modified using a vector to deliver a working copy of the faulty ABCD1 gene. And here, the vector that's carrying this healthy copy is introduced or mixed with the stem cells, and that transduce these cells or insert the healthy ABCD1 gene into the stem cells. Then the patient undergo a myeloablative therapy to make room for the modified cells. This is ex vivo gene therapy, meaning the cells are removed from the patient and then altered. This approach is not appropriate for AMN. Therefore, we are working on an in vivo treatment for AMN. It is on the right side of the slide here. AAV9 vector carrying the working copy of ABCD1 gene is injected directly into the CSF, the fluid that surrounds the spinal cord and brain. With this injection, AAV9 vector will deliver the functional copy of the ABCD1 gene to the cells of spinal cord. And here is the vector that's carrying the healthy gene and is delivered. This figure is not exactly accurate, but it's delivering in the spinal cord, not in the arm, into the CSF. And then from there, the vector delivers this gene to the cells of the spinal cord. With cell delivery, that's with the Bluebird approach, each patient is treated with a unique product each time. Each patient's treatment is developed via a multi-step process, as I described above. Direct delivery, that's appropriate for AMN. So direct delivery with a single dose of an AAV gene therapy is not only more convenient for patients, but it does not involve all the room for error with a multi-step process. In addition, the products are developed for all patients versus each patient on an individual basis. Now I'll walk you through how AAV delivers ABCD1 gene to the cell of the central nervous system. In this photo, an AAV vector is shown. It carries ABCD1 gene and vector gene to deliver the payload to the nucleus. And this is a vector itself. On the bottom, you can see the cassette that has the ABCD1 gene. And here, let's do the next slide. And you can see here has the ABCD1 gene and a few small genes to help this vector insert that into the nucleus. So this vector carries the ABCD1 gene to the nucleus, where it delivers it to nucleus. After the injection of AAV, the AAV is endocytosed, and will now this is a structure of a cell. In the cell, it could be a spinal cord cells or in the brain cell. When AAV is injected, it is endocytosed here into the cell. And then it's transported in the cytoplasm within this structure to the nucleus. And then only the gene enters the nucleus and form an episome in the nucleus. And you can see episome here. It is not inserted in the genome of the cell, but it stays outside the genome in the nucleus as its independent episome. This is the final destination for the gene. From here, the gene is translated and transcribed to produce healthy ABCD1 gene, and that process is shown here. Once the healthy gene is in the nucleus, then as needed by the cell, new ABCD1 proteins are produced. And this protein, when it's produced, is transported to peroxisomes. This is a one-time therapy. The patients can receive this therapy for one time only, and they should see all the benefit with one injection. And it's very important because the lowest dose that we are going to deliver, or we plan to deliver in patients, we want to make sure that has an efficacy and it's an efficacious dose. No one should receive a gene therapy product which is not efficacious just because this is their one opportunity to receive it. And I'm going to talk that more when we talk about our non-clinical studies. In this slide, what I'm showing is that AAV9 gene therapy has been studied in clinical trials for past 2 decades. This is not a new modality. Over 3,000 patients have received these therapies. Two AAV therapies have been approved and are commercially available. An AAV2 gene therapy for an eye disease and an AAV9 gene therapy for children with spinal muscular atrophy, or SMA. The AAV9 used for the SMA is similar vector to what we are using for AMN. For SMA, the drug product delivers the gene needed for SMA patients, and SBT101 delivers the ABCD1 gene needed for AMN. A team in Boston tested the AAV9 gene therapy in mouse model for ALD. After the treatment, the healthy ABCD1 gene was observed in the cells across the whole spinal cord, confirming that vector can deliver the gene to the cells of CNS. And this led to reduction of very long chain fatty acids, and it's depicted on the right side of the slides, where you can see the wild types has normal very long chain fatty acids compared to the knockout mice, where it's higher, and you can see the dose dependent reduction. You see a reduction in ABCD1, very long chain fatty acids with AAV9 vector. This is not a multi-dose effect depicted here, it's just different routes of administrations. But you can see that gene has been delivered and it results in reduction in very long chain fatty acids. This work prompted us to develop the gene therapy for AMN patients. Before I move on to that one, I want to point out that the inserted gene not only reduced the function, it also improved the function. So the treated knockout mice retained function compared to untreated knockout mice. And their function was very comparable to wild type mouse. And this function was measured with hind limb extension scores, or um, the hind limb function. So the gene was reached the right cells, was able to reduce very long chain fatty acids, and that resulted in improvement in function. We replicated the work but using much higher doses. Graph on the right, let me look at it, on the left side of the slide here is depicting the level of human ABCD1 gene in mice. In wild type, we're not expecting to have them to have a human ABCD1 gene. And as you can see in here, as we move from left to right, the doses increases. With increased dose, we see higher amount of ABCD1 protein. Y-axis is amount of ABCD1 protein. Higher it goes, the higher amount of gene we have. And that's what we expect with the higher dose of the vector. We confirm that gene is delivered, and to investigate if it works, we check the very long chain fatty acids, and levels were reduced in spinal tissue and significantly reduced in the high dose group at 13 and 24 week. And we can show that is shown here on the right side of the slide. The x-axis, the y-axis is very long chain fatty acid levels, and x-axis are the four groups. First group, as you may remember from the first slide, is wild type placebo. Second is placebo, and then low dose and a high dose. And you can see that there's a dose dependent drop or decrease in very long chain fatty acids in these mice who are treated with the gene therapy carrying the healthy ABCD1 gene. As I said earlier, the patient can only receive the therapy one time using today's technology. We want to make sure the patient in low dose and high dose receive a dose that has a proven efficacy in animal studies. This is different when compared to traditional therapies, where a patient can start with a lower dose and dose can be increased to improve its effectiveness. This approach doesn't work for gene therapy. Hence, we're doing a lot more studies to confirm that the low dose that we test is efficacious, and patients enrolled in the low dose arm see a similar level of efficacy. If not, it should not be less than the other doses, because this is their one-time chance to get treated. Our program have two types of studies. Our program in humans have two types of studies. Treatment study, where we will investigate the AAV9 gene therapy, is one, and has not started yet. We are completing animal studies needed to start the first in human, needed to determine the first in human dose, and a requisite to start the human study. Once we're ready to enroll in our treatment studies, I will reach out to you and inform you. I'll share the update with you. The second study is the natural history study, which helps us understand the progression of the AMN disease. This will also help physicians and regulators to decide about the efficacy and safety of the gene therapy. This study has started earlier this year. In Europe, the study is ongoing in Amsterdam and Germany. Dr. Lackman's and Dr. Elaine Murphy's site in London would very soon will be fully active and start enrolling in UK too. If you want to join this study earlier than that, you can reach out to Amsterdam or Germany, and they can help you enroll in this study. In the US, the clinics in Stanford and Boston are ready to enroll. In addition, we, in collaboration with Autobahn, are developing patient-reported outcome measure with the University of Rochester. This will be an AMN-specific endpoint that can be used in the future trials of AMN. It will not only be for a gene therapy study, but it can be used for all treatment studies by all companies. This outcome measure will be for both men and women. So as you may have noted, the existing outcome measures like 6-minute walk test that was developed for lung and heart disease is not the best for AMN. And we need an AMN specific outcome measure, and this can be the one of that first one. To develop this PRO, or patient reported outcome measure, we need your help. This is a survey that all men and women with AMN, yes, women with AMN or women with ALD should complete. More surveys we get, the better it is, because this has to meet the regulatory authority criteria. And if you have not completed that survey yet, please look for the link and complete your survey. I know Sarah and Karen and their team are helping us to distribute that link to all of our participants here. I'll stop. Thank you. And if you have any questions, I think we'll have a question sessions live, and I'll be happy to answer any question that you may have.